everyone to Osteobites. Please feel free to add questions. And my name is Christina Itoma, and I am mom to OsteoAngel Dylan and Director of Scientific Programs at MIB Agents. Today on Osteobites, we're talking with Dr. Kurt Weiss and Tanya Heim from the University of Pittsburgh. Dr. Weiss is a 2021 Outsmarting Osteosarcoma grant recipient, and they will together discuss his funded work on exploring aldehyde, dehydrogenase, and androgen receptor actions to outsmart osteosarcoma metastases. Thank you so much, Dr. Weiss and Tanya, for joining us on Osteobites today. We're thrilled to have you. And thank you to Vicki for joining us as our panelist today. Vicki is an osteo warrior and MIB Agents Senior Advisory Board member. There are many ways that I can introduce one of our guests today. Dr. Weiss is our 2021 Outsmarting Osteosarcoma grant recipient, as I mentioned. He's also an osteosarcoma survivor, and he's a skilled orthopedic oncologist at the University of Pittsburgh. He's also a native Pittsburgher who has been involved with research since 1994, when he performed orthopedic basic science research as an undergraduate student. And today he's an associate professor of orthopedic surgery in the University of Pittsburgh School of Medicine, Department of Orthopedic Surgery, Division of Musculoskeletal Oncology. He also directs the department's Cancer Stem Cell Laboratory. And in addition to his clinical practice in pediatric and adult musculoskeletal oncology, he directs a basic science laboratory dedicated to the study of sarcomas. He holds a Career Development Award from the NCI to develop a translational sarcoma research program at UPitt. And as a bone cancer survivor himself, he brings passion and enthusiasm to the laboratory, clinic, and operating room. And through UPitt's Cancer Institute, he is also a proud collaborator with several scientists who are trying to understand how other forms of cancer, particularly breast cancer, spread to and destroy the bone. He's a founding member of the Musculoskeletal Oncology Research Initiative, the Pittsburgh Cure Sarcoma, and the Pittsburgh Center for Bone and Mineral Research. And we're also excited to welcome Tanya Heim today. Tanya has a BS in Animal Sciences from Penn State and a Master's in Biology, Immunology concentration from Youngstown State. And she continued her study of stem cells by exploring their use for hernia repair in rat models in a graduate biology program at Youngstown. And following graduation, she worked at UPitt in a liver transplantation research group and breast cancer research lab, where she developed murine mammary tumor xenografts. And she taught anatomy and physiology at Utica College and advanced physiology courses at Colgate University. And she continues to teach online, but she moved back to Pittsburgh in 2019 to continue her cancer research with Dr. Weiss's lab at Pitt. And she's a technical project lead for the project that is funded by the MIB Outsmarting grant. So very excited to have both of you here today. And before we jump into your presentation, we just have a few announcements. In addition to our Osteobite series, MIB Agents offers many programs for the osteosarcoma community, including Warrior Mail, where osteo warriors receive monthly care packages with personalized letters and words of encouragement. Patients can sign up to receive Warrior Mail, or you can sign up to be a Warrior Mail writer. We'll drop a link in the chat where you can sign up or to receive or write. And all MIB Agents programs are free of charge, thanks to generous donors who make it better with us. And there are a couple more ways you can support MIB Agents in our programs now and in September for Childhood Cancer Awareness Month. We are still doing Outrunning Osteosarcoma. You can run, walk, cycle with us now through September. You can choose from a 5K, 10K, half marathon. You can rack up your miles anytime between now and the end of September. It's a great way to raise awareness for pediatric cancer and show that you care. We'll put a registration link in the chat. And also during September for Childhood Cancer Awareness Month, you can make and sell gold mailbox bows in your community, or just purchase a bow to raise awareness for pediatric cancer. And we will put a link in the chat where you can either order a bow, or if you're interested or know someone who'd like to get involved in making and selling bows, we'll put some contact information in the chat as well. And then lastly, before we get started, I want to thank the sponsor of this episode, BTG Specialty Pharmaceuticals. BTG provides rescue medicines typically used in emergency rooms and intensive care units to treat patients for whom there are limited treatment options. They are dedicated to delivering quality medicines that make a real difference to patients and their families through the development, manufacture, and commercialization of pharmaceutical products. Their current portfolio of antidotes counteracts certain snake venoms and the toxicity associated with some heart and cancer medications. Their drug Varaxase is for high-dose methotrexate toxicity. All right, Vicki, you want to go ahead and introduce yourself?
Yeah, hi everyone. So as Christina already mentioned, my name is Vicki Hoy, and I am a junior advisory board member at MIB and also an osteo warrior. I'm also a current freshman at Villanova University studying finance, and I'm so thankful to our guest joining us today. So if you guys would like to take the floor.
Thank you, Vicki. Thank you for that, and Christina for those kind introductions. I wish my mom could have heard that. Thank you all for having us again. We are sorry that we missed you at Factor. I heard it was amazing, predictably, and we are very, very grateful to speak with you this afternoon about what we've been up to in the laboratory since we were last together. So what's gone on since the MIB posse descended on Pittsburgh to present our award, which was marvelous? Well, not much, just that we've had a major personnel change. Dr. Rebecca Waters, who had been my scientific right hand and co-director of the lab, has moved on to an industry job where she hopes she continues to do good, and we wish her the best, obviously, but we sure miss her. We have changed our venue. We moved the cancer center, or our laboratory, from where we had been up to the cancer center, which is where we are now. That move took place about a month ago. And the reason that is significant is because instead of having a 20-minute drive from where we look after osteosarcoma patients in the clinic and operating room, well, now we have a 4-minute walk. And that is a big enough deal in itself. But when you got a prosthesis, a 4-minute walk is much better than a 20-minute drive. So we're very happy about that move. Closer to our collaborators, closer to the patients, closer to the science. It's really going to change things. So with that, let's get started in what we've been up to in the laboratory. Osteosarcoma is the reason why we're all here, to make it better for our patients with osteosarcoma. This is a disease that has an incidence a little bit more than 1 in a million. There will be 500 to 700 cases in the United States this year. This is a Dalenogram named after Dr. David Daleen, who was at the Mayo Clinic for 1 million years. And this is a wonderful picture. Can you see my mouse? That is fabulous. This is a wonderful picture because it tells you who gets it, so mostly guys, when they get it in the first and second decade of life, and where they get it. So distal femur, proximal tibia, proximal humerus, the most common sites for osteosarcoma. And this is what we are obsessed with in the Musculoskeletal Oncology Laboratory. Just about all metastasis is to the lungs. And we're going to talk about this a lot throughout the talk. When my patients get into trouble is not because so much of the tumor in their arm or their leg. I've got a pretty good answer for that in the operating room. We don't have as good an answer about what to do with the osteosarcoma after it's spread. And so that's what me and Tanya want to own. We want to be experts at the biology of how osteosarcoma cells can do that. How does a cell make that difficult journey from the, starting in the distal femur here and crawling out of the tumor, going through the bloodstream, recognizing you're in the lung, crawling out of the bloodstream into the lung and start growing, all the while chemotherapy is trying to kill you? How does a cell do that? That's what we need to figure out. Before we had the audacity to give children and young adults chemotherapy for osteosarcoma, we didn't do a very good job. Survival was around 15% 5-year survival. And that changed marvelously with the addition of adjuvant, meaning before surgery. neoadjuvant before surgery and adjuvant chemotherapy after surgery for osteosarcoma. And that was almost overnight between, I'll say, around 1965 and 1985, this wonderful change happened. And that's great news. The bad news is we haven't improved those numbers in certainly as long as I've been looking after this disease or had it as a patient. And we're doing as crappy for patients with metastatic disease as we've always done. So what does that mean? Well, this is Katie, and I diagnosed Katie and operated on her in 2015. She is now a rising junior at West Virginia University, and she's going to be a marvelous nurse. And Katie's prognosis in 2015 is the same as mine was in 1989. And I know that there's nobody who can hear my voice right now that thinks that's okay. It's not okay. And just to put a exclamation point on it, this is a axial CT scan of the lungs of a sarcoma patient. And the impressive thing about this scan is not the large bone sarcoma on the inner table of this patient's left scapula. I can take care of that. In fact, I did take care of that with surgery. What I was powerless to do anything about, and what eventually killed this 34-year-old father of three, was this tremendous burden of metastatic disease in the lungs. All these white dots here, these large round spots, those are all sarcoma metastases. And that's why I'm here, and that's why Tanya's here. We must do a better job of understanding the biology. And once we do that, we can figure out how to stop it. ALDH stands for aldehyde dehydrogenase. This is a factor, I know you like that word, a stem cell factor that we've been interested in for about a decade. And what ALDH does, it's ALDH 1A1, we see here, is one of the members of this family that has like 14 members to it. Basically, this is a drug or an enzyme that allows cells to withstand stress. So if you think about that journey that we just outlined, that the cell takes from the distal femur to the lungs, well, that's a pretty stressful journey for a cell to take. So a cell that's going to make that journey successfully while chemotherapy is trying to kill it, it had better be able to withstand a lot of stress. So we thought, well, maybe this ALDH enzyme is important. Also in the literature, my friend Kanya Hinoki from Japan suggested back in 2010 that ALDH might be important for osteosarcoma resistance, and other publications have correlated ALDH activity with metastatic potential in human and mouse osteosarcoma cell lines. We've done some of that work. We've done a lot of that work. And Matteo Trucco, who is another Outsmarting osteosarcoma winner, and I have a great deal of interest in this thing ALDH. So okay, if let's put our money where our mouth is. If it can't be just theoretical, right? It's got to matter. It's got to be important. So these are two human osteosarcoma cell lines that are related. SALS2 has been around a long time, and a lot of osteosarcoma investigators use this enzyme. And what my mentor and hero, Jeannie Kleinerman at MD Anderson in Houston, did was she took this cell line and she cycled it through the lungs of experimental animals. So she took SALS2 and put it in the tail vein of experimental animals, and some of them got metastases. And she took that metastasis and put it back in the leg. And then it went to the lungs, and she took that lung met and put it back in the leg. And then that went to the lungs. So what she did very brilliantly was select out a very highly metastatic variant of this guy here. So think of this cell line as just this cell line, but it's really good at metastases. So not metastatic, highly metastatic. Okay, you get the picture. Well, when we look at the expression of the ALDH 1A1 gene in SALS2, it's quite low. In LM2, it's quite high. And this recapitulates what we saw in mouse osteosarcoma cell lines with differing metastatic potentials as well. Anybody who's listening to this talk has heard the name Lee Hellman. Well, Lee Hellman has a couple of cell lines named K7M2 and K12. They are also related. One is not very metastatic and one is highly metastatic, and they have the same relationship. This picture is superimposable if you call this K12 and you call this K7M2. Okay, and when we look at our patients at the University of Pittsburgh, our sarcoma patients who got metastases and those who didn't, if we look at their ALDH expression, we see that there's a pretty dramatic difference. So we really think that this thing, ALDH, matters in terms of osteosarcoma metastatic potential. So another experiment that we did that we're hoping to try and publish soon is that we looked at a bunch of primary osteosarcomas, that's all these tumors under the green bar here, and a bunch of osteosarcoma metastases under the red bar here. And we did RNA sequencing on them to see what was different. Was anything different? So I had always thought that they have got to be different, but until you prove it, you didn't prove it. So we did the experiment, and lo and behold, we can see on this RNA sequencing heat map that the gene expression of these guys, the primary tumors and the metastatic tumors, is pretty dramatically different. Not shocking, makes a lot of sense, but again, until you demonstrate it, you didn't demonstrate it. And one of the things that we found that was really exciting is that when we looked at these two groups of patients, the metastases and the primary tumors, we found that ALDH was overexpressed in the metastases, which was very exciting because this was unbiased, meaning you can't fudge the results, you get what you get. And to find that an enzyme that we were interested in studying for like about 10 years prior to this was showing up yet again was very exciting and made us very happy. But that's not all we discovered. We found in doing pathway analysis that perhaps one of the things that was driving ALDH expression was this guy, the androgen receptor. So who cares? Why is that important? Well, if you remember back to that delenogram, which we showed a few slides ago, mostly dudes, but obviously not always, right, Vicky? Get osteosarcoma. So it's been conjectured for a long time, and when you look at when kids get, and by the way, women have androgen too, they just convert it to estrogen. That's not a part of this talk, but that is a biological phenomenon. And if you look at when kids get osteosarcoma, typically, again, not always, Ann Graham, but mostly, when kids get osteosarcoma, it's their peak of growth velocity, right? The first and second decades of life. So it's been conjectured for a long, long time, boy, sex hormones got to have something to do with this, seeing who gets it and when they get it. But it's never been proven. And so to find that androgen receptor, this thing that we've surmised has been important in osteosarcoma biology for a long time, but have never really proven it, might be driving ALDH and other things. Well, that was pretty exciting too. And again, we hope to publish these data before too long. And finally, before I, and this is what we just talked about. So again, who gets it are young people and in the peak growth velocity. So this is why we thought a lot of people, including my hero, Dr. Henry Mankin, who operated on me, thought that androgen receptor and androgens might be important. And if we look at these data from the Broad Institute, if you look at androgen receptor expression by cancer subtype, way the heck out here on the left with the highest expression is osteosarcoma. Now there's got to be a there. There's got to be something to that. And so our application was to try and dive into that biology a little bit of androgen receptor and osteosarcoma metastases. And what the heck is going on with ALDH? What's that all about? Is androgen receptor somehow driving this? Is one more important than the other? You know, who knows? So the point is to try and drill down to this biology a little bit. And why does it matter? Well, we have drugs that inhibit ALDH. We have drugs that inhibit androgen receptor. These are both what we call actionable targets, right? So if this is something that we've got to, we ought to be adding to the armamentarium of osteosarcoma treatment to try and finally budge that 65% number and do something for kids with metastatic disease, well then we need to do it. So that's what our project is about. And at this point, I'm going to let the real brains of the operation in to explain what we've been up to in the laboratory. So Tanya, you ready?
Yeah.
All right.
Hey, Dr. Weiss, I had a couple questions actually.
Yeah.
Slide, you know, when that showed the AR in the different disease types.
Yeah.
And I'm curious for some of the diseases that are further along that end where there is greater AR expression, do those cancers also typically happen in the same age group?
So that is a great question. So let's just go through a few of them. So GCT, giant cell tumor, is absolutely a disease of younger people. Not quite as young as osteosarcoma, but still young. People typically are in their 20s and 30s. So there is some overlap. It's actually a great question. And also interestingly, giant cell tumor is one of the very few things that I treat, like osteosarcoma, that also has a male predominance. So that is, there's probably a there too. Prostate cancer, you know, pretty obvious, only dudes get that. Chondrosarcoma tends to be a disease of older people. Chondrosarcoma is a little bit weird to get at someone Vicky's age. Although it happens, it's just not typical. Glioma tends to be a disease of younger people. So, and here we are with Ewing sarcoma, right? We all know who gets Ewing sarcoma. Same people get osteosarcoma demographically, you know, young people in the first and second decade of life. So that is, Christina, that is a very astute observation, and one that I must confess I did not appreciate until you brought it up. You know, I just focus in on this bit. I wasn't looking further down here, but a smart person like you would do that. So thank you for pointing that out.
Someone who doesn't know very much would, but I really like the colors on the chart. So actually, one more question, sorry, before you move on, and maybe actually, can you go back to the slide that showed the RNA-seq? So yeah, so if I'm just understanding this correctly, so you're finding high ALDH expression in the metastatic cells. I was just trying to think if there was any implications for risk stratification, but is it that you're not, even if there's tumor that's likely to be highly metastatic, would you not see that ALDH in the primary tumor yet? Is it, you don't see it until it metastasizes and then it's in the...
Another really terrific question. So we actually did that work, and Tanya's going to be the lead author on this paper. So I'm going to put you on the spot a little bit. We did break out the primary tumors, the ones that eventually had experienced the clinical event of metastases, and those that did not. And did we see a difference there?
Most of the time they clustered separately. Primaries. So, sorry, I'll come on there. Most of the time when we looked at the characteristics, the genetic characteristics between a patient, so some of these patients, we had primary and metastatic tumors. So we could compare, do those primary metastatic tumors, are they more like each other? Or is that net more like another patient's met, or is that primary more like another? And so we did that. We don't have this data on here, but yeah, we did that, and we did see that the primary tumors did, for the majority, cluster with other primaries, and for the majority other metastatic tumors clustered with other metastatics and did not have a relationship to that patient. Yeah. I think the only one, and we ended up, I think, eliminating it from our study, was the extraskeletal myxoid case, or
extraskeletal osteosarcoma.
Yeah. Extraskeletal osteosarcoma case, and that one was a little strange, in which the primary and metastatic tumors were more alike, but this is not your conventional osteosarcoma.
Okay, so you wouldn't necessarily see these markers in the primary tumor until it starts to metastasize.
Yeah, it seems that way.
But the interesting implication there is that if you look at the primary tumor and the metastasis from the same person, like I had metastatic disease. So if you look at my lung met has more in common with other kids' lung mets than it does with my own primary tumor, which is fascinating. And with that, I'm gonna and terrifying. And with that, I'm going to give it over to Tanya.
All right.
So this brings us, with all this background, and I'm going to give you a little background as I go through the results too. Not really maybe the most standard way of doing this, but I think maybe easier of understanding our results if I just give methods with. But so with everything that Kurt talked about with ALDH and AR, we did come up with a hypothesis that if we overexpress or knock down these two genes of interest, the ALDH and the AR, in osteosarcoma cell lines that we already have been working with and tested, two different osteosarcoma cell lines that have different metastatic potential. We believe if we overexpress them or knock out these cells, that we'll be able to actually figure out the best treatment option for patients specifically with metastases. So how do we do this? So this is something that has been a process over the past year. So we have some of these cell lines created, not all of them. One thing, did you mention that we moved? Okay, I couldn't remember. So with our move, I have basically half of these cells created, and we're working on some more data with those cells at the moment. And then I have some other cells to create. So basically the cells that we have that we've created are some knockdown cells. And so basically we wanted to take these two cell lines, SOUS2 and LM2, and knock down these genes of interest, ALDH, AR, and then also the combination of the two genes together. So we took SOUS2, this is a primary cell, osteosarcoma cell, and it's just a commercial cell. And so it has very low metastatic potential. It's not a very aggressive cell. And so this is a cell that we have tested in the past, and we saw that the ALDH does have lower expression in SOUS2, and ALDH has higher expression in this LM2. This is a graph that Kurt just previously showed. What is also interesting is SOUS2 has higher AR expression. We did not show that in this talk, but SOUS2 does has higher AR expression than LM2. So they're kind of flip-floppy. ALDH high in LM2, but AR low in LM2. And in this one, ALDH is low, but AR is high. So we know their base genetic makeup, at least concerning these two genes. So we take these cell lines, and then using a virus, we actually infect the cells. And I actually have this on another slide showing you kind of how we do that. So I'll go through the process of how we do that. But the end point is that we want to knock out ALDH in SOUS2, we want to knock out AR in SOUS2, and we want to knock out both in SOUS2. And we want to do that also with the LM2, the more metastatic cell line. The other thing is overexpressing these genes. So again, want to overexpress ALDH, want to overexpress AR, and want to overexpress both of them together in these cell lines. So it took a little while to figure out exactly how to do that. It's not like you just think about, oh, you aren't able to just do that without some back work. So first thing you do is you have to actually make these vectors, these lentiviral virus. Basically lentivirus, so it's a derivative from like the HIV virus, and you make these, you put these vectors into the virus. And so I won't go through all of these things, but basically the things that I want to point out is the important things for these cell lines is we want to create an ALDH cell line that if ALDH is overexpressed or knocked down, that these cells, whenever we look at them under a fluorescent scope, we'll see a green color. And for AR cells, when we transfect them or infect them with these lentiviral vectors, if AR is overexpressed or knocked down, we will see them fluoresce a red fluorescent protein. Now we have different, lentiviral vectors for overexpression and knockdown. So that's how we'll know which expression it's at those cells are actually showing us, because I'll know exactly what virus I transfected in. Our gene of interest would be in this, what we call a short hairpin loop. And so this is part of that lentiviral vector. And this is actually what would get encoded into the DNA of our cells going forward. So I just wanted to kind of point out that, we have these fluorescent proteins that are markers for us to easily look at these cells under the scope as we're going along and make sure that they're giving off the right fluorescent protein, just kind of as a check along the way. But the thing that we had to do some back work on is finding the promoter for these genes. So we can't just ask the cells to either start overexpressing or expressing less of a certain gene. We actually have to target that gene by targeting an area before our gene of interest, which would be called a promoter. So they have some nifty things now. Over the past, I think 5 years, maybe, or 5 to 10 years, they've come out with some really nice ways of selecting your best promoter. So they have, so what we did was actually worked with a company called Horizon, and they provided us with this smart choice promoter plate in which they have a series of different promoters or none in this 96 well plate. And you can see that they basically, so what's in these plates is the promoter at different dilutions. You can see from 8 all the way down to 0.25, and then a duplicate to make sure that what we're seeing is actually truly what we're seeing. In our case, this duplicate actually was helpful because we were able to find the best promoter for SOUS2 and the best promoter for LM2 in one plate. So what happens here is these promoters are in these plates. We seed cells into this 96 well plate. Some other work that was done that's not shown on this slide was we figured out the best transduction media. So this is our media that the cells grow in, plus or minus sometimes, but most of the time plus a chemical called polybrine that actually will help loosen essentially the membrane, the proteins in the membrane, so that you can actually have a virus slip into the nucleus a little easier. Typically, you don't want too much because then you're just starting to dissociate all the proteins of a cell and you may kill your cell. But we figured out a pretty nice concentration of polybrine that seemed to work with transfection of our cells. So we use this polybrine transduction media and then incubated our cells with our lentiviral particles in these particular wells with a promoter. And then after 6 hours of transduction, essentially with these, we stopped transduction and then we imaged these plates for up to 96 hours, starting at 48 and then every 24 up to 96. So what we saw with that is when what you're seeing here is not, again, it's not, so it is a GFP, fluorescent protein like we had talked about that would be on a virus, but these are specifically associated with the promoter that you see here off to the left. Now I'm only showing 2 of all of these promoters of the 8 or 7 different promoters that we tested, because these are the 2 that really worked the best overall. So I figured that's like the best comparison. And then of course, empty, just showing that we truly did not have anything show up. There was no, false negatives or false positives, sorry, or anything like that. So what you're seeing here is for SOUS2, the less metastatic potential cell. At 48 hours, we imaged the cells from that plate. At 72 hours, we also imaged the cells of that plate. And at 96 hours, we again imaged the cells. And over time, I just put little stars here to kind of indicate what we thought at those points, what were the best promoters or and the best the best dilution of those promoters as well. So we saw right away that this is human cytomegalovirus. It's a very common promoter for these types of lentiviral transduction experiments, but there have been a lot of papers recently where we've even seen like the best promoter for sometimes human cells can even be mouse. So that's why we include even mouse. That's what the M would be for underneath it. But we did see human CMV did work the best for SOUS2, and it was consistent all the way through. My stars got moved a little bit, but we saw it was consistent all the way through 96 hours. And so definitely wanted to go with HCMV for our promoter as SOUS2. And nicely, doesn't always work out like this in research, but nicely, we also saw the same thing for LM2. And I'm just showing the 72 and 96 hours here for, just so we don't have to go through all of those pictures for LM2. But yeah, so we saw that HCMV was our promoter of, choice that we wanted to go forward with. So we made these lentiviral particles. We ordered these lentiviral particles, I should say, and got them in. And then basically once we had these, we were able to transfect our cells. And so far, like I said, we have used the knockdown lentiviral particles to knock down ALDH and knock down AR in both the SOUS2 less metastatic and LM2 more metastatic cell lines. So this is kind of what it looks like. What the vector essentially would look like now. So a little more background on some experiments that we've done, and then I'll get to like the nitty gritty really of this particular project. But so speaking of SOUS2 and LM2, we have looked at both of these cells with single treatment, as what I'll call them just for sake of this talk. I'll call, if we're only using disulfiram or the other drug would be enzalutamide, I'll say single treatment, or double treatment would be with both. So we did look at disulfiram treatment alone on our cell models, and these are not knockdown or overexpression cells yet. These are just the base cell lines that, again, where SOUS2 would have low ALDH but high AR, and LM2 would have high ALDH but low AR. So disulfiram is a ALDH inhibitor. It's an FDA-approved drug already, and this is something that we've been using in the lab with a lot of our previous ALDH research. Unfortunately, so we did see that LM2, the more metastatic osteosarcoma cell type here, it does seem more sensitive to disulfiram alone than does our primary osteosarcoma cell line. But you do have to use, we weren't seeing these results consistently significant all the way through. We actually had to dose these cells for 3 days in order to see these results. And so it took a while for us to actually see the sensitivity that was there with these metastatic cells, but we did see that that LM2, the more metastatic cell type, is more sensitive to disulfiram alone. And so this is on I should have explained this too, I'm sorry, on the y-axis is cell viability. So 100% is our max. And basically we just tried different treatments or tried different doses of disulfiram. From untreated vehicle would just be our vehicle that we mix the drug in. So no drug in vehicle, just the solution itself. Methanol would hopefully kill everything, just like if you drank a bottle of methanol, that most likely is not going to do your body well. And then we have these varying concentrations along the way. And I will point out that our lowest concentration here that you see is 50 micromolar. That'll come into play later when I show the combination therapy. So we did the same thing with SALS2 and LM2 in treating those cells alone with an AR inhibitor, enzalutamide. So enzalutamide, again, is an FDA-approved drug that a lot of prostate cancer patients are currently using. So it's something that was of interest because it's not a new drug, and so we might be able to get this, might be able to use this more regularly in the clinic. What we saw was that we didn't have as stark difference in enzalutamide treatment as we saw with disulfiram. We were thinking, okay, LM2, the more metastatic cell line, has really high AR compared to the less metastatic cell line, SALS2. So we thought that inhibiting that AR might significantly reduce LM2 cell viability. But again, if you see here on the left, cell viability is on the y-axis and our different treatments are along the x-axis. And there was no significant difference between the two cell groups until we really got to really high concentration. So that wasn't too, that didn't give us too much confidence yet, but like we said, we thought there was a link between ALDH and AR. And so we wanted to move forward with our combination therapy treatments as well. And again, this was treatment for 72 hours, not just a day or anything like that. So next we looked at cell viability of our two cell lines. The one I'm showing you here is LM2, and then we'll go to SALS2 next. So LM2, remember, the more metastatic cell type. When we treated LM2 with enzalutamide and disulfiram, what we did was we used enzalutamide at what's called the IC50 concentration, and then so IC50 means that this is a dose concentration in which you give to a plate of cells and 50% of the cells die. And so then you know like what that dose is, and so then you're not over treating the cells, because if everything dies, that's not really telling you much anyways, and if nothing dies, that's not telling you much either. So you want that kind of sweet spot of 50% of the cells are resistant and 50% of the cells are sensitive, so you know that at least the drug is getting into the cell, and that's kind of where our baseline start is that IC50. So then we take then we know that enzalutamide's IC50, and we start doing serodilutions from that. So different percentages of that IC50 dose was tested, along with different doses of disulfiram, all of which are fairly low dose compared to their IC50 as well. What we found was the combination treatment of enzalutamide and disulfiram. We found the most significant reduction in cell viability with this double treatment. Right here, where we were treating the cells with enzalutamide at only around 6% of the IC50 of enzalutamide. And then this graph here on the right is actually basically if we take this boxed in area and turn it into a bar graph. So again, treating all of these cells with around 6% of the IC50 of enzalutamide, and then we have these varying concentrations, all again reduced from the IC50 of disulfiram. Which of these combinations actually gives the most significant reduction in cell viability? And we saw with LM2 that the most significant decrease was actually all three of these, which was kind of exciting, I'm not gonna lie, because not only do we have one option to treat with this combination therapy, but now we have several. And what's also nice is when you have 3 different ones to choose from, you can always choose the lower drug concentration, because obviously we want to try to get the drug doses that we're giving to be as low as possible. So going forward, we were really interested in what would be this green bar group of treatment of the 6% of IC50 enzalutamide, and then it's 1 micromolar of disulfiram. So a lot less than what we were seeing previously. So that was for LM2. For SOUS2, we did the same exact thing. We treated with different concentrations of enzalutamide compared to, again, they're all reductions of the IC50 of enzalutamide for SOUS2. And then we also treated with 4 different variations of disulfiram. So we saw that the most significant reduction in SOUS2 cell viability was actually at our highest concentration of enzalutamide. It was at the 100% IC50 of enzalutamide. And also, when we looked closer at that, it was also the most significant reduction, actually, as a similar significance of, so basically we had all 3 of these groups significantly reduced for LM2, and to find the same significant reduction for SOUS2, it was only this one group. And this one group for SOUS2, we're giving a much higher dose of enzalutamide and also a higher dose of disulfiram. So that was interesting because normally we think of SOUS2 as being the easier cell to treat, I guess, in our mind, because it's the primary cell and we might have more answers for that, as Kurt said. And normally we see our more metastatic cell types resisting drug therapy in the lab and more consistent with what we see in the clinic as well. So this was really interesting for us because we're seeing SOUS2 be a little more resistant when we actually do combination therapy. But also obviously very exciting to see that we have options for metastatic therapy. So that was cell viability. And the other part of our project is not only to look at cell viability, but the, kind of the meat and potatoes of the project is we really want to make sure that we're not only killing these metastatic cells, but we're decreasing their ability to migrate. That's really the thing that we want to stop, is their migration, their spread, their metastases. And so we ran some migration experiments with SOUS2 and LM2, treated and untreated. And we did treatment with enzalutamide alone, and then treatment with enzalutamide and disulfiram at their specific concentrations that seem to work best for both those cell lines. So we treated LM2 with this treatment here, the 6% IC50 of enzalutamide, and 1 micromolar of disulfiram. And then for SOUS2, we used its most significant treatment, the IC50 enzalutamide and 2.5 micromolar disulfiram. So using their most significant treatments, we ran migratory tests, like I said, with the IC50 of enzalutamide for these, and we saw that enzalutamide did significantly reduce LM2's migration. So LM2 untreated versus LM2 treated. We did see that there was a significant reduction just with the enzalutamide treatment alone. And this again was for migrated cells. We saw less migrated cells with enzalutamide treatment. However, we saw, now again, this is at the IC50 of enzalutamide, and then we saw a similar significant reduction of migration of these LM2 cells with that reduced IC50 enzalutamide and disulfiram. So this is again the 6% IC50 and the 1 micromolar of disulfiram, which is I think like 50% of the disulfiram IC50. So at these severely reduced concentrations of doses, we see the same significant decrease in migration. So that's really exciting that we can relatively decrease these doses to treat these more metastatic cells, but we're seeing the same reduction that we would see at higher IC50. And personally, I think we're excited about that because enzalutamide, we don't really want to inhibit androgen receptor completely in children that are going through puberty. That could, as I think anyone could think of, could have some side effects. We don't really want to block that pathway at that particular point of time for a child. However, if we're able to reduce the activity and still maintain and still be able to have that child maintain normal normal metabolic, go through more normal metabolic processes that come with growth spurts and things like that, then we might be able to get away with treating with an AR inhibitor in these young individuals if we keep the doses down. And so that's really what the goal of all of this combination therapy at the moment. The goal first was to see if does it work, and now we're getting greedy and want to see, okay, how low can we go and it still work?
Basic question, but like, how do you look at or measure cell migration when it's?
No, that's a great question. I didn't put it in here, and that's so it's good that you asked. There's actually some really nifty plates essentially that companies sell. And so we just take a 24 well plate, so a plate with 24 wells, and down the middle of that plate in 2 rows, there are these little inserts. They almost remind me of, it's gonna sound really weird, but if you have ever done like communion with the little grape juice cups or something like that, so those little tiny cups, they basically look like little inserts like that, and you put them in the wells. But the bottom of those little cups are mesh. So they allow easily for media to mix and vitamins and other like small amino acids, things that are very, very small or aqueous, can get through that membrane easily. Things that are larger, like cells, so we just grade that mesh so that if a cell wants to get through that membrane, it must actually go through that membrane. It must actually physically put out extensions, pull itself through, and get to the other side. And so what we do is we put these little inserts in the plate. We plate our untreated or treated SALS2 and LM2 on top. And then we put in the bottom either, this is where you see FBS positive or FBS negative. What that means is in the media underneath these inserts, they either have media that they like, which would be the FBS positive, or media that they don't necessarily like, which would be FBS negative. Basically, FBS is fetal bovine serum. And you normally put that in any media that you're culturing cells with in the lab. But the big thing about it is it has a lot of growth factors. It has a lot of yummy nutrients for the cells. It is all the things that you can get out of the plasma that are very nourishing and necessary for cells. And so this is what you put in your media, and this is what they like to grow in. Almost all cells, unless they are manipulated a certain way to not grow with FBS, almost all cells need FBS to grow and maintain their cellular functions for what's expected. Normally, if you take FBS away, cells are not going to be attracted to that area. So that's why our FBS negative wells, those are basically distinct negative controls for our groups, just to make sure that we don't have increased migration in these FBS negative areas. Now, we did see with LM2, they really like to migrate no matter what. And so we did see the FBS negative or negative control groups just as high or higher than groups that had that chemo, that had the FBS to attract them to migrate. But we honestly think that this is just because LM2 is such a highly migratory cell that it doesn't, from when you grow LM2 versus SALS2, SALS2 will grow out in a cell, in a flask, and love to be right up against other cells and touching all these other cells. And it doesn't matter how many cells are in there, just keep growing and proliferating until they're basically on top of each other if you let them. LM2, it grows fast, but it likes to grow far apart. And so that's one thing with like why I think also it doesn't matter if we have a chemoattractant in these migratory experiments with LM2, though we will do it because it is, scientifically like necessary. But I don't know that that LM2 cares, because their main goal is to grow out and they become these really large cells compared to cells 2 and have these long extensions. And they really just try to get to, if I'm growing a flask of LM2 and I put a colony of cells in one spot, in a day or two, all those cells will have dispersed from each other and wanted to go to different places in the flask. They don't really like to be next to each other. They want to move. So that's why in these negative control, or why I think in these negative controls, why they're maybe, I don't think they're necessarily telling us that our experiment didn't work. I think they're telling us something more about LM2. And then, and I did not explain HT1080. I'm very sorry. HT1080 we used as a positive control. HT1080 is a what's been described as a dedifferentiated chondrosarcoma. So not an osteosarcoma, but we know that this cell loves to metastasize. We know that this cell loves to grow really fast. So we knew it would be a great cell to use as a positive control in this, because again, and we do see similar things with HT1080, because it loves to metastasize, there wasn't a significant difference between whether or not we have a chemoattractant to attract those cells to migrate or not. But yeah, very good question. Hopefully that answered everything.
Completely, thank you.
Okay. Okay, so moving on from this, I'll go through the conclusions really quickly, and if there's any other questions, we'll get through them. So conclusions that we found so far with our study is that LM2 does seem to be more sensitive to disulfiram treatment alone. However, I'm actually going to jump down to the last bullet point because I feel like I probably should have put it up higher. However, LM2 is actually also seems to be more sensitive to AR inhibition with the addition of disulfiram. So it seems like, because if you remember, LM2 was not more sensitive to AR inhibition, so was not more sensitive to that enzalutamide treatment alone. We didn't see significant decreases in cell viability when we treated with the AR inhibitor alone. But when we combine it with disulfiram, we're able to get really significant decreases in cell viability and also in migration. So that's LM2. Seems to be more sensitive, which is what we want. But we did see that whenever we treated LM2 alone with enzalutamide, we didn't see those significant reductions. So the most significant decrease that we saw in LM2 cell viability was with doses much less than the IC50 of either drug, enzalutamide or disulfiram. However, SOUS2, our less metastatic cell, we're actually having to use larger doses, which for this study, we're not concerned with, but it is an interesting thing to see that actually it's probably a good segue when we were talking about this heat map and about how primary samples seem to cluster with other primary samples, and metastatic samples seem to cluster with other metastatic samples. It's really interesting to see the very different ways in which these two cells are being treated. We're seeing, instead of SOUS2 just doing the same thing that LM2 is doing, it's actually very different. So that makes me also believe our sequencing data even more, if I hadn't already. But obviously we're not done yet, so we have a good amount to still keep chugging along here and doing. So we're going to do a couple of validation, a drug therapy migration experiments. So basically what we want to do is take those knockdown and overexpressed SOUS2 and LM2 cells, either having AR or ALDH knocked down or having those two overexpressed in these two different cell types and doing those migration experiments. And actually, I should be starting those for the knockdown cells next week. So that's really exciting. So I'm excited to see what I'll get with that, because yeah, it'll be all those knockdown cells with and without treatment. And then we'll be able to kind of see how that correlates with our not old, but this data, these data. And then, so we're going to be doing that. We're also going to be validating our cell lines through PCR to make sure that those genes are genetically reduced. And then we're also kind of in the same vein, while we're running these migration experiments and we're treating cells with different drugs, I've also been collecting and will continue to collect RNA from all these different cell lines. And then we're going to send those off sequencing so that we can actually look at all these particular genes of interest and see if there's any differentially expressed genes that are either overexpressed or underexpressed in these two cell types, and see if that tells us anything more, especially with how we want to treat these drugs. Do we want to specifically target the gene, or is our method of like an oral medication more promising? So sequencing will help us determine that as well. All right, and then, yeah, do you want to give all the thank yous? And
right. So just to wrap us up, thank you, Tanya. That was awesome tour de force of what we're trying to accomplish in the lab. Just want to thank my whole team. It's not just me, it's not just Tanya, it's a whole lot of people, an army of osteo warriors that helps us. Want to say a few words about this guy. So Luke Carlson is another osteosarcoma survivor. He had an osteosarcoma of his proximal femur and was looked after at the Mayo Clinic in Rochester, Minnesota. And Luke got himself a job at Pitt, and also got into a post-graduate program, a post-baccalaureate program, and just got himself into the med school at Pitt. And he just had his first couple days of classes like Vicky did. And this is me and Luke at Luke's white coat ceremony. So 2 generations of osteosarcoma survivors, pretty cool.
That's very cool.
Yeah, so obviously thank you to all our funding sources, especially MIB agents making it better. And yeah, so this is in our old lab before we moved, as we were packing everything up. One of the people who came out to visit us in Pittsburgh was Allie, and I think it's pretty safe to say that she stole all our hearts away, and we love her. So we're thinking about her, and thank you again for this opportunity.
Thanks so much, Dr. Weiss and Tanya, for joining us on OsteoBites today and making it better for osteosarcoma patients everywhere. More information on this and all OsteoBites can be found on YouTube, on our website at mibagents.org, and at your favorite podcast place. And please join us on OsteoBites next Thursday, September the 1st. We're going to be talking to Dr. Robert Cantor and Dr. Robert Rebhun from UC Davis about their study on inhaled recombinant human IL-15 in dogs with naturally occurring pulmonary metastases from osteosarcoma or melanoma. It's a phase 1 study of clinical activity and correlates of response. And you can find our OsteoBites lineup for the rest of 2022 on our website. And if you have any ideas for future topics you'd like to hear about, please share them with us. And I dropped an email address in the chat. Thank you again, Dr. Weiss and Tanya and Vicky, for spending an hour with us today, and to our sponsor, BTG Specialty Pharmaceuticals. And thank you all for joining us today on OsteoBites. We hope to see you next week, and really appreciate the update, Dr. Weiss and Tanya. I know you're kind of at the halfway point, and so it was really great to get a peek behind the curtain of everything that's been going on. And we look forward to talking to you again, like maybe in another 6 months or so.
Awesome.
Sounds good.
Sounds
All right.
terrific.
Thank you guys. Bye.